So now I'd like to bring to the podium Dr. Bailey Arruda. She comes to us today from the United States Department of Agriculture, Animal Agricultural Research Services. She's a research veterinary medical officer there, where she studies the interactions between influenza A viruses in swine. And she's going to give us an update on that today. Take it away. Forward pointer.
Oh,
back.
awesome.
Stop. Don't ever press that.
I will press it now, but you told me not to press it. All right, well, good afternoon, everybody, and thank you for your time this afternoon. I'm going to be touching a little bit of background on highly pathogenic avian influenza, touch briefly on our issues with swine-adapted influenza in the United States, what we've been doing in our inoculation trials to assess the risk that high-path avian influenza poses to our swine herd over the last few months, and then where we hope to go in the next couple years to further evaluate these different strains. And then I'm going to end the talk hopefully telling you what we think we know about these viruses in pigs so far. So highly pathogenic avian influenza is an influenza A virus, meaning that it has a segmented genome. So if two different influenza A viruses infect the same cell, you can get reassortment. This is an incredibly complex situation, and so we cannot necessarily predict what will come out of those two parental strains in terms of their progeny. In general, it could lead to increased fitness within the host. It is a defined phenotype in poultry, in gallinaceous birds, leading to high mortality due to the systemic distribution of the virus. It does not mean that it is highly pathogenic in humans, in cattle, or in swine. And unfortunately, right now, we cannot predict which virus will pose the most risk to livestock based on sequencing. So lucky for us, multiple adaptations are required to overcome species barriers. In general, the first one that are seen when an avian virus jumps to a mammal is the BB2 mutations within the polymerase. So most commonly, these are seen to be E627K and D701N. These mutations allow the virus to interact better with host proteins and replicate the virus within those cells more effectively. The next jump that occurs in general is going to be that avian influenzas prefer an alpha-2,3 sialic acid linkage compared to mammal influenza viruses that prefer an alpha-2,6 sialic acid linkage. So there'll have to be a jump to a different receptor linkage. Unfortunately for us, pigs and people do have a similar distribution of alpha-2,3 and 2,6 within their upper respiratory tract and their lower respiratory tract. Beyond this jump, then the HA-NA, there has to be a balance between those two different surface proteins. So the HA binds and the NA releases. So those things have to play nice together so that the virus can get through the mucus layer and then back out of the cell and back through the mucus and to the next host. Unfortunately, reassortment may overcome many of these barriers in one round of replication. So highly pathogenic avian influenza, there's a new paradigm, obviously. It is a panzootic. It has now circulated across the globe with all continents, being detected on all continents other than Australia. It does appear that the virus has adapted to wild ducks and migrating waterfowl, so it looks like it might be with us for a while. There is evidence in Europe that it has become enzootic. Additional to this adaptation in migratory waterfowl, there are unprecedented amounts of mammalian infections with this virus. Multiple introductions have occurred within North America, except an early European introduction seemed to have stuck and has remained and has developed into America's subclade right here. There is frequent reassortment with North American low-path avian influenzas that have led to co-circulating genotypes with potentially different phenotypes. With very few exceptions, this H5 has been paired with an N1. So this slide is really just to show you that it's more of a them than an it. We often talk about highly pathogenic avian influenza as a singleton, but really it's numerous different viruses. They all belong to this HA clade here, but they're further classified into genotype based upon if it's wholly Eurasian versus reassortments. And then there's an additional reassortment that has come up in the last few months that aren't actually on this next table. They're then further categorized by segment group. I'm not entirely sure, and I don't know if anybody really is, at what level does the pig care about all of these differences within genotypes. High-path avian influenza genotypes do wax and wane. So you can see a time scale here from the beginning to July. And this is across different species groups. And you can see the different colors by time, by different genotype. If you look closer in the last year, the back is going to be your poultry, or your wild bird, my apologies. The bar is going to be your poultry, and then you can see the cattle. So you can see in the last few months, a lot of detections have occurred within poultry and cattle of this B3.13. The CDC is monitoring different wastewater sheds and has been able to detect high-path avian influenza within different states, within different watersheds, including Colorado, Minnesota, Texas, California, Idaho, as well as Arkansas. More than 10,000 wild birds have been infected, and when you look at the strains that are being detected within these birds, very few of them have mammalian adaptation markers. If they are present, it's an E627K, and it's often thought that these birds are eating infected mammals, and this is why they have that mammalian adaptation marker within the virus that killed them. So in mammals, many, about 20%, have mammalian adaptation markers. When they're present, it's E627K above the other ones. There are more domestic cats, mouse, and rabbit and voles being tested, and those are commonly associated with dairies. More than 100 million gallinaceous birds have been infected, with 10 known associated human cases within the United States. Again, very few of these strains coming out of these poultry species have a mammalian adaptation marker. When they are present, it's commonly found in those birds that are located on backyard farms. Cattle, 14 states. Obviously, I didn't update this, last night, so it should be 214 cases, 4 reported human cases associated with that, with maybe 1 more in Missouri. What can be sequenced from that case right now looks to be very similar to the cattle strains, but there is no known exposure to cattle. Looking at those sequences from the cattle, they do have a BB2 M631L mutation, which is thought to help with mammalian adaptation. And they also have a mutation within the HA that's thought to increase pathogenicity within humans. We have done some bovine H5 research within the NADC of late, and so I just touch on this because I think it provides some information that would be impactful for swine producers to think about. So as Dr. Scanlon said, the bovine mammary gland appears to be a great viral factory. You're getting virus out of these girls that is in so much logs greater than you would get from the nose of an infected pig with a swine-adapted virus. So there is concern for untreated waste milk and where that goes and what happens to it. In looking at the pathology, the loss of the secretory alveolar basement membrane as well as fibrosis suggests milk production will be affected in the glands. And then when you feed milk from from these girls to bucket calves, they do develop mild clinical signs. You do get RT-qPCR detection in the upper respiratory tract, and they do seroconvert. So it's not overly surprising that the logs of virus that are coming out of these girls is infectious to calves. So I just want to touch on this publication from 2015 in which they took the pandemic H1N1, gave that intranasally to kit ferrets, and then they were able to detect it in the ferret mother's mammary gland, her milk, and by IHC in the histopath from those girls. So it isn't necessarily in dairy cattle the first time that we've been warned that it can cause mastitis and transmit. As you guys all know, swine-adapted influenza infection in the US is common in commercial production. And not only that, but because of reverse zoonosis, human-adapted gene segments in swine influenza viruses are present, which increases the risk for reassortment. So not only could reassortment with a swine-adapted influenza increase the fitness of that high-path avian influenza in swine, it could also increase its fitness and ability to replicate in humans because there are human-adapted gene segments within our swine population. So our pathogenesis and transmission studies look a bit like this. The first 4 strains we tested were in this study design. We then expanded the study design to look a bit more like this when we started to understand that these viruses didn't play like the other viruses that individuals at ARS had played with in the past. So we expanded our study design to look at different tissues within the pig, as well as different sample types. When you look at the next table in the next slide, I just want you to keep in mind that so far we've done weaned pig models. We use clean pigs. They don't have MHP, they don't have influenza and immunity, they don't have currently circulating influenza in them, and they're seronegative for PRRS, PCR negative. These occur in a BSL-3. So there's 6 whole room air exchanges per hour within the facility. Temperature is controlled, and it's low humidity. Pig numbers are relatively low. There's just no way we're going to be able to replicate the hundreds and hundreds of pigs that are in a barn. We have no pit sample, which I think could be unfortunate because perhaps a pit sample could act more like a bulk tank sample for farms. The good thing out of these studies is that we also get known status samples so that we can use those in the future to develop and evaluate more rapid diagnostic serologic assays or different sample types, meaning oral fluids or diaphragm for meat juice. We will be looking at muscle in this next study where we're looking at the B3.13 viruses within pigs. So we're expanding that study design to include muscle, diaphragm, as well as 0, 3, 5, and 7-day whole blood PCR to look for viremia. Oh. All right, so this is the summary slide. There are some similarities and some very important differences between these strains in pigs. This is an oversimplification of the differences. And so if you want to get more granular, you will find more differences than what is illustrated in this table. You can see on the side there's strain ID. And so the turkey and the bald eagle did not have any adaptation markers. They did replicate in the lower respiratory tract. There was no nasal detection. There was no transmission. Clinical signs were not evident. They did have lung lesions at necropsy, and they did have histologic lesions. However, the number of pigs with lung lesions did vary, and the severity and antigen distribution did vary by strain. We did not look for anything outside of the respiratory tract in the first study. If you look at the raccoon and red fox, they both had the E627K adaptation. We saw replication in the lower respiratory tract, nasal detection. However, it's not red. Well, it's not red because it's inconsistent nasal detection by PCR, and it's pretty high CT values with these strains. Transmission, we did see transmission with these 2 viruses. Clinical signs were not noted. Lung lesions, again, were observed. The most recently performed study looked at these 3 strains. All of them came out of turkey vultures. They all had different adaptation markers, but they were all of the same genotype. So they all replicated in the lower respiratory tract, and nasal detection was consistent across these viruses, except this turkey vulture, Wyoming, with the D701N, had pretty significantly different nasal detection than the other 2, meaning that it shed more similarly to a swine-adapted virus. So all primary inoculated pigs did shed it on consecutive days at decently low CT values. We did see fecal detection out of all of these last 3 viruses, And why this is red is because there was actually more fecal detection in that turkey vulture California than there was nasal detection. Again, the CT values are pretty high, and it is fairly inconsistent, however it was present. We did see transmission in 2 out of those 3, but again, this E627K didn't actually transmit compared to these 2 that did. Mild clinical signs were seen in terms of respiratory and increased fevers that were really very short. However, in this D701N, we saw neurologic disease in these pigs. So 4 out of the 20 primary inoculated pigs did develop neurologic disease that included stargazing, circulating, and then if you would take one of the pigs and put them on their back, they would not right themselves. They also had seizures. So looking at detection outside of the respiratory tract in these last 3 strains, we were able to detect it in the brain in the first 2 strains, being the turkey Wyoming and the turkey Utah, as well as the pancreas in all 3 of the last strains. So looking at the images below, you can see this is the kind of the classic appearance of what would be a swine-adapted influenza, multifocal patchy pulmonary consolidation. This is the bald eagle, that's NPIHC, and that's an endothelial cell within the lungs. This is also from a bald eagle pig where the antigen distribution is pretty extensive compared to what you would expect for a swine-adapted influenza. This is the brain from one of the pigs from the D701N. Unfortunately, when you start looking at the H&E of these guys, you could think that it was a chronic bacterial meningitis because there is inflammation within the meninges that are lymphoplasmacytic, histiocytic inflammation. So you need to take a little bit closer look to the neurons and see if they are actually checking out or necrosing. This is an ethmoid from one of the 701N pigs, and you can see here, based upon the location, it appears that the virus can infect the neurosensory cells of the ethmoid, and then it seems to like it tracked down into the axons subjacent to that, so it's a potential entry point for this virus to go through the nose to the ethmoids and to the olfactory bulbs. This is NPIHC with replication of the virus within the pancreas of the pigs. And this is the duct within the pancreas, so it does look like it likes to at least focus on the bile ducts for replication. We have looked at vaccines. We took an Astrakhan mRNA vaccine, and we challenged with red fox isolate that was on the previous table. It did look like it protected, so lucky for us, if you get a good match for the HA, you generally get pretty good protection with influenza vaccines. So these guys had less, the vaccinated group compared to the positive control had less virus with their lung based on PCR CT value as well as IHC. We have looked at knockout pigs. So Dr. Baker has a collaborator at Missouri that can produce ANP32A knockout pigs. And so we challenged those with the D701N strain. And there is still testing to be done on this study, and we're actually running a replicate of this study next month. But it does look like the wild-type pigs did develop neurologic disease like the other study, so it is reproducible. The thought process on this is based upon the fact that in humans, it does appear that the ANP32B protein is more proviral, so you will have this mutation, while in pigs, it looks like the ANP32A protein is more proviral, and so you'd have a mutation that looks like more like this, and which would be more fit within the pig. We're looking at feral pig serosurveillance in those guys with the National Pharaoh Swine Surveillance at USDA. We've run about 4,000 sera, about 3% are NP positive, and less than 0.1% are H5HI positive. So, and a lot of these, a subset of these, a decent number, I would say a few months worth of testing has occurred after the cattle outbreak. We're looking at lectin staining and virus histochemical staining. So we're working with Iowa State with this. Iowa State on this. So this is cattle, and from this publication from Iowa State, in which they looked at 2.3 versus the 2.6 in the mammary gland of cattle. So remember alpha-2,3, avian, alpha-2,6, mammalian, and you can see the cattle express those very nicely within the secretory alveoli compared to the ducks, where there's some differential expression. So this is from the mammary gland of a sow. You can see that the alpha-2,3 avian isn't really that well expressed, while the alpha-2,6 mammal, it is expressed within the secretory alveoli, within the intralobular ducts, and within the interlobular ducts of the mammary gland. So you can pair this with virus histochemistry. So this is virus histochemistry of an H1N1 from a pig in a lung, just as a demonstration. So what you do is you take the virus, you inactivate it, you label it with FITC, you put it on the slide, you see where it binds, and you detect where it can bind. So here you can see that this H1N1 attaches nicely to the respiratory epithelium, as well as what I think are PIMs within the alveolar septa. So what does the next several years for us entail? Well, we got a good amount of funding from the USDA because they do think that the highly pathogenic avian influenza does pose a risk to swine, and so they want some additional information. So we have been tasked to look at additional strains. Our next study will start in October looking at B3.13 strains. We're looking at 3 different strains with different markers in them, as well as a representative strain. We'll look at another D701N from a cougar at some point, as well as the new C genotype. We're hoping to get into additional models, looking at the sow model, and then looking at a pig to ferret transmission study, as this is considered the gold standard to understand the likely transmission from pigs to humans. We're starting the paperwork to look at reassortment so that we can better understand what's going to happen if it gets into a herd with the currently circulating influenza. We are likely to start at least with the most frequently detected clades, and after that start selecting based upon what we think will be good matches or mismatches between the polymerase as well as AJ-NA pairings. We'll also look at impact of prior influenza immunity on shedding characteristics as well as pathology. Vaccine evaluation is on the list, of course, and then develop or optimize assays and sample types for sensitivity and specificity for detection of high path in swine. Specifically looking at more rapid serologic assays. An HI, H5 is fairly labor intensive. It requires a BSL3 or a deselected H5, and so making something that can differentiate between an H5 and H3 and an H1 would behoove the industry, I do believe. And then looking at something that can be easily collected, such as oral fluids, might help in surveillance efforts as well. So what we think we know so far. Pigs are at risk for the North American H5 circulating strains, potentially put it on your differential for CNS disease. CNS disease wasn't seen in contact pigs. However, again, we're only working with 8 contact pigs and 20 inoculated pigs, so we're not going to be able to get to the numbers that could potentially tell you if it's an uncommon occurrence in contact pigs. Reassortment with the endemic swine strains is a concern. It might lead to increased fitness within both pigs as well as people. There is experimental evidence as well as evidence from the field that if low-path avian influenza reassorts with a swine-adapted influenza, it's more able to transmit within swine. Risk of incursion is likely low. However, avoid untreated surface water, ensure bird proofing, restrict scavenger mammals, don't feed untreated milk to swine. I was recently on a call where they did a survey of some dairy producers, and a small subset, a very small subset, did feed some of milk to swine. So just a big call out there, please don't do that. And then evaluate the biosecurity risk posed by dairies. And you're going to have to do that within your system, thinking about how much virus is actually in the milk and if you share trucks or share personnel with dairies. So there's a long list of people involved in all of these studies. It takes a village to run these studies in BSL3, so thank you to all of them, and thank you for your time.
Stay nearby. Okay. Questions for Dr. Arruda while we're switching up? John Korsland has a question.
Contact swine, if you have pigs in contact with infected cattle or infected pigs, or
No, yep, in the pathogenesis and transmission study, what we do is we infect primaries via intranasal inoculation, and then 2 days later, we'll put contact pigs in to see if it transmits.
So you infected pigs or infected cattle?
We infected pigs, so pig to pig.
Okay, where's the data on the infected pigs with the B1313?
At the end of October, I'll have some of that
Oh.
for you.
You haven't done it yet.
Nope, so they're going to be moved into the BSL-3 beginning of October and inoculated
Not
like October.
to be too critical, but it's 6 months, and that's a very critical question. What's taken so much time?
Well, we are a small group there, and we have been doing cattle studies.
Yeah, I understand. But
So,
it's pretty critical for pigs, too, especially the sow question, I think.
nope, yep, so we're on it. We're trying to do it as fast as we can.
One more question, Dr. Snyder.
Hi, Tim Snyder, PSC Europe. I was wondering if you've considered the fact that pigs are often fed whey from the cheese process. Would you consider that to be a risk? I'm not sure if whey would be, it's quite acidic, so I'm
Okay.
not sure if the virus would survive or not, but just a thought.
So I don't have a lot of knowledge based upon the processing of whey. If there's a process, is it pasteurized before it goes into whey?
I don't know. Yoshi did that work.
Okay.
Yeah, UW Madison did that work, Yoshi, lab, and so we can pull up that paper. I think it's still in preprint, but he separated the milk, sorry. UW Madison, Yoshi, who's one of the grandfathers of influenza research, separated the milk and all the different particles, parts, whey, skim, and whatever, and he did all the virus. And I don't remember, but the virus was in different amounts depending on the part of the milk that he was looking at.
Yeah, I used to have a couple of finisher bars that were built close to the cheese factory.
Yep.
So and they had pipeline.
And we're very efficient in animal production of using products that would be considered waste to find full value in them. But it's time to reassess that. What's the risk versus the reward of using a product that may or may not have virus in it? And how to ensure that. So I'll get that Yoshi publication out to you. It's still in preprint. So thanks, everybody. One last round of applause for Dr. Arruda as she exits the stage.